This topic lecture covers one of the three main structures of the cytoskeleton, the actin filaments. Here are the topic objectives for this lecture. In this lecture, we are going to cover the basics. What are actin filaments? What functions do actin filaments perform? And lastly, what controls actin filaments? Let's start by looking at the structure of the actin filaments. These actin molecules are the tiniest of the fibers that make up the cytoskeleton. These fibers look like twisted chains of actin subunits and have polarity, just like microtubules. Just like microtubules, actin can assemble or disassemble, and this process is entirely related to the molecule associated with the actin molecule. If ATP is associated with the actin, the subunits have a strong bond and stay associated. If the ATP is hydrolyzed, then the bond becomes weaker and the actin molecule is more likely to disassemble. This also creates the polarity that we saw in the microtubules. We are going to talk in a moment about all the different functions of actin within the cell. But how does actin perform all these variable functions? This has to do with the actin-related proteins known as ARPs. Each ARP allows a different structure to be built, so to speak. The first function of actin is to form the structure known as the cell cortex. This is a meshwork of actin right below the plasma membrane in the cell and helps provide the shape and the support for the cell movement. One of the main functions of actin is to support cell crawling. This action is important in immune cells, which must patrol our cells for foreign invaders. There are three steps in cell crawling that are supported by actin. The first is polymerization at the plus end that helps the lamellipodum to protrude out of the cell. The second stage is where the protrusions adhere to the new ground being covered. This provides traction for the cell. The last step of this process is where the rest of the cell drags itself through the contraction to the protrusion. Through these repeated steps, the cell can move forward. Remember, it is only through the ARPs that are functioning in conjunction with actin that the cell is able to crawl. There are, of course, other molecules that are involved in the attachment and traction of the cell, such as integrins, but we are focusing on the role of actin. If you would like to see more on cell crawling, feel free to check out your textbook. Myosins are the name of the family that actin-dependent motor proteins belong to. These motor proteins are similar to the ones we discussed in the microtubule lecture, but in this case, myosin is specific to actin, and these two proteins work together. Depending on where myosin binds within the cell, this can help determine the shape of the cell. So what controls the arrangement of actin? This happens through extracellular signals that the cell receives. In this case, the Rho family protein is primarily responsible for the arrangement of cells. This all goes back to when we talked about cell communication. Depending upon the signal that is received, along with other internal and external signals, actin will either be polymerized or not, and the action will determine the shape and action of actin within the cells. That's it for this lecture on actin filaments. Please review the objectives for this topic and let me know if you have any questions.